Hi, good morning. Welcome to the second day of SPAOM. And let's start this new session on superresolution. Today, it's my great pleasure to introduce Jonas Ries. Jonas is a group leader at EMBL at Heidelberg. And his group, Superresolution Microscopy for Structural Cell Biology, uses bespoke and state-of-the-art imaging approaches to explore nanoscale multiprotein machineries in their native and functional cellular contexts. Jonas is very well known for the amazing microscopy approaches he employs, and also for the beautiful biological results he obtains from them. Today, Jonas will tell us about elegant and comprehensive work his lab did to create reference standards for virtually all superresolution approaches. It's fantastic. This is based on nuclear pores, and he also delved a bit into his efforts to increase throughput and speed in single molecule superresolution microscopy. Jonas, the floor is yours. Thank you.
Thank you so much, Pedro, and welcome everybody. Good morning. Let me share my screen and we can get started. Good. So my name is Jonas Ries, and my group at the EMBL in Heidelberg is interested in understanding how complex protein machines are organized in cells. And for this, we are developing new tools for super resolution microscopy. So the technology that basically all our work is based on is single molecule localization microscopy, and you might also know this under the acronyms of PALM or STORM. So this technique has been introduced quite a lot already during this conference. So I just want to briefly remind you. So in single molecule localization microscopy, what we need to use are fluorophores that we can switch between a bright state and a dark state. Then in the beginning of the experiment, we switch off all the fluorophores, and then we acquire thousands to sometimes millions of camera frames. In each of these frames, we then switch on a very small subset of the fluorophores, because then these single fluorophores are well separated. We can determine their precise positions, and from these positions slowly fill in the super resolution image. So this super resolution image then can have a resolution that is 1 order of magnitude and sometimes even 2 orders of magnitude higher or better than the normal diffraction limited image here. So to remind you, the quality of localization microscopy is mainly determined by 2 factors. The first one is how well can we localize the positions of the single fluorophores, and this is determined mainly by how many photons we detect. So we want to have bright fluorophores. The other is the labeling density or the labeling efficiency. Because we have such a high resolution, we really have to make sure that we decorate our target structures very, very densely with our target fluorophores. And this can often be the limiting factor. So today I want to very briefly start with showing you how we do 3D and multicolor single molecule localization microscopy, and then move on to a topic that is quite close to my heart, and this is how we can do quality control in single molecule localization microscopy. And then in the second part of this talk, I will tell you how our efforts in my group to increase the speed and the throughput in single molecule localization microscopy. So when we perform our localization microscopy measurements and localize the positions of the single fluorophores, we get from this process usually only the X and Y coordinate, but no information about Z. And this is a problem because biological structures are intrinsically 3-dimensional. And for many biological questions, we just need to know where the proteins are in 3D. That's why quite early already, localization microscopy has been extended to 3D. And the idea is to not only look at the positions of the single fluorophores, but also at the shape of the images of the point spread functions. This is usually done after introducing some aberrations, in this case, after inserting a cylindrical lens that leads to astigmatism and these kind of elliptical point spread functions. And then by just looking at how these point spread functions look, what's the aspect ratio of these ellipses, we can calculate back Z. So there's many variations of this approach, but what they all have in common is that they're quite robust and also easy to implement, but also that the Z resolution is not perfect, and especially in the beginning, the Z resolution was not as expected, not as we hoped it to be. And one reason for this limited performance was that during the analysis of our 3D super resolution data, we had to assume that the point spread functions were described by Gaussian functions. But realistic PSFs often are not Gaussian, and this, of course, then creates a problem. And this is why in my group, we now developed a new fitting approach that uses not Gaussian points and function models, but experimentally derived and therefore much more precise points and function models for fitting. And these PSF models we can obtain from stacks of beads immobilized on a cover slip. So now we can resolve, for instance, here these immunolabeled microtubules in the side views as these beautiful hollow cylinder-like structures. So there's still one limitation of this approach, and that is that we get this high resolution only in a relatively thin slice around the focus. In this case, it's 500 nanometers. We might be able to push it to 1 micrometer, but that's kind of the end. So what can we do if we want to do 3D localization microscopy in entire cells or tissue? Well, we don't have to take only one slice in Z, but many slices one after the other, and then later stitch them together. What is nice is that in each of these slices, we already have a high 3D resolution. So we don't really lose resolution when we do this. So this we did here, again, for microtubules in a cell, and you see here this rotating reconstruction. And also below here in this kind of single slice that moves through the cell, that we can really achieve a quite good resolution. However, there is one problem, and in the beginning, when we tried this approach, we couldn't really use it. And the reason is that we could only acquire a few slices before losing all the flow force. Because whenever we take data in one slice, flow force in all the other slices already get bleached. So this data we could only take after discovering that one dye, CF660C, turned out to be very photostable, and that we can activate it many times, switch it on many times, and switch it off many times. This allowed us to then take 20, 30 slices one after the other without an apparent loss in localizations. And I think this is quite useful. So we can get such nice data in 3D, not only in a single color, but nowadays up to 4 colors. And our approach to multicolor localization microscopy is to use only 2 spectral channels and then use fluorophores that are spectrally quite close and that we can see in both channels. So for normal microscopy, this would be a disaster. That would be horrendous crosstalk. We couldn't tell the signals apart. But because we do single molecule localization microscopy, we know that we're always looking only at a single fluorophore. And therefore, by just looking at the intensity ratio of the single molecule event in both channels, we can precisely determine the color, the identity of the fluorophore. So what you see here is one nuclear pore complex where we labeled 4 of its components. And above here, I now show you an average calculated from about 200 nuclear pore complexes. And this now quite nicely shows you where the individual proteins are in the complex. So now I want to talk a bit about quality control, because this is something that I think was really missing in the field. And that made it very difficult for us to determine if our microscopes were as good as they were a year ago, or as good as they are as the microscopes of another lab somewhere else. For example, you see here 2 publicly available data sets showing microtubules, both taken with kind of the same technique, single molecule localization microscopy, but these images don't look exactly the same. And the reason for this varying quality can be actually manifold. It can be due to the microscopes, due to the imaging conditions, due to the labels we use, the sample preparation protocols, and even the software we use for the analysis. And to test all these points and to optimize them, what we really need is a sample that is the same everywhere and every time we look at it. And this is a standard sample. So we now teamed up with a group of Jan Ellenberg at the EMBL and generated such a standard sample. And our idea was to use for this the nuclear pore complex. This is the channel between the cytoplasm and nucleoplasm. And what is nice that here nature positions proteins at well-defined 3D positions in a precisely defined stoichiometry. So we chose as a target protein NAP96 that forms 2 rings in the nuclear pore complex. Each of these rings has 2 × 8 proteins. And what is nice about this protein is that both rings are in register, so that in the top view, we can nicely see the 8-fold symmetry of the nuclear pore complex. We then created homozygous CRISPR cell lines in which we labeled NAP96 with 4 commonly used labels. And that means that in these cell lines, all of the target proteins are labeled. So we don't have any unlabeled proteins. And since we label the endogenous proteins, we also don't change the number of proteins in the cell. So after we generated these cell lines, of course, we wanted to try them out, and we wanted to just look at them with all the different kinds of microscopy modalities we had access to. And this you see here. And this is actually quite nice because now we can directly compare different microscopy modalities on precisely the same sample. You see here that with the diffraction limited techniques, we can resolve individual nuclear pores quite nicely, but of course, we don't see any internal structure. So with stat microscopy, another super resolution technique, we can nicely resolve these hollow rings. And these rings are also visible when we use expansion microscopy, and that was collaboration with the Enriquez group. Now, if we turn to localization microscopy, first with these rather dim photoconvertible fluorescent proteins, we again see this nice ring-shaped structure, and we start seeing this 8-fold symmetry. And this 8-fold symmetry is beautifully visible when we use the much brighter organic dyes. Now we can ask questions such as, can we resolve these two corners of the nuclear pore complex? Or in Z, can we resolve these two rings? Because then we know we have a Z resolution of at least 50 nanometers. So for us, however, these nuclear pore complexes turned out to be mostly important to optimize the entire sample preparation and imaging workflow. The reason that they are so well suited for this is that the samples are very robust, reproducible to prepare, and also quite robust. And also that in a single focal plane, we can have thousands of nuclear pores in focus. And this gives us a huge statistical power and allows us to even see minor changes in the quality if we change certain parameters in our workflow. But also very importantly, the nuclear pores allow us to measure the labeling efficiency. So that's the probability that a target protein has a label attached to it that we can later see. And this is a parameter that otherwise is actually quite difficult to determine. But with the nuclear pore complexes, it's quite easy, because if you don't have 100% labeling, then we won't see all the nuclear pores as complete rings, but corners start missing. So our approach is to automatically segment hundreds of nuclear pores, then automatically count the number of corners that have a fluorophore, a localization event, and then to fit a probabilistic model to this histogram of the number of corners. And this fit directly gives us the absolute labeling efficiencies, and this with a quite high statistical accuracy. So this is useful, for example, to test the reagents that you use in your lab. In our case here, the nanobodies that we generated ourselves. So this is a good batch of nanobodies against GFP. This is a bad batch of nanobodies, and now we can put numbers to it, how good or bad our nanobodies are. And it turned out that even our good batch of nanobodies went bad after 2 years in the fridge. So now we really use this assay to regularly test if our labels are still acceptable. Now there's something more we can do with these cell lines, and this might be more interesting for biologists, because these cell lines allow us to count the number of proteins in a complex. And the stoichiometry of a complex, of course, is important for its function to understand its structure and so on. So counting in single molecule localization microscopy means that we have to relate the number of localizations to the number of proteins. So these numbers are not exactly the same, because on one hand, we might have limited labeling or limited maturation of our fluorophores, and that leads to undercounting. On the other hand, basically all the fluorophores that we use have a tendency to be activated not only once, but also maybe a second time, and this leads to overcounting. Now, there's one approach that is insensitive to these two artifacts, and this is counting using a counting reference standard. Such a reference is a complex with a known stoichiometry that we label with precisely the same label as our unknown complex and image under identical conditions, because then we can directly calculate the stoichiometry of the target complex from the relative number of localizations. So our nuclear pore cell lines turned out to be very well suited as these counting reference standards, because they are very easy to segment. So nuclear pores never overlap. Therefore, we would never segment two nuclear pores as a single nuclear pore. Also, they have this characteristic shape and size. And because of this, we would never take some background dirt, background noise, and think it's a nuclear pore complex. So we now validated this counting by counting the number of NAP1 or 7, which is another protein in the nuclear pore complex, and for which we know that the stoichiometry is also 32 copies. And indeed, we hit this number spot on. And if you now look at the standard deviation of repeated measurements, you see that's below 10%. And I think this is quite good for intracellular counting. But we don't need to use even super resolution microscopy to perform counting with these nuclear pore complexes. The reason is that even in a normal confocal image, as you see here, we can resolve individual nuclear pores. And that directly tells us how bright 32 GFDP labeled proteins are. And with this, we then can convert these grayscale images with arbitrary units into precise images that directly show the protein concentration. So again, we validated this with NUP 107. And as before, we again achieved a high precision and also a high accuracy for these counting measurements. So to summarize this part of the talk, so we generated these homozygous cell lines where we labeled NUP 96 with commonly used labels. And these are quite useful to determine the microscope performance and resolution, to optimize imaging conditions, including labeling efficiencies, and to count the number of proteins in a complex. So we deposited these cell lines on a public repository, the CLS, and already quite a few groups are using these cell lines, and I think they are slowly becoming the gold standard for quality control in super resolution microscopy. And if you're interested, just try them out, either to check your microscopes, but also to just get pretty pictures for your next publication. So now I want to switch gears and talk about speed and throughput in single molecule localization microscopy. So single molecule localization microscopy is really an annoyingly slow technique. It takes minutes to hours to acquire the 10,000 to sometimes even 1 million camera frames that we need to reconstruct a single super resolution image. This, of course, is not ideal if you want to perform high throughput measurements, for example, to compare many different conditions or to find rare events. And it is even worse for live cell localization microscopy, where the speed of the process that we're interested in dictates how fast we have to go. So to overcome these limitations, in my group over the years, we tested several approaches. On one hand, we automated our microscopes to have them run around the clock and therefore produce a lot of data. Then we tried to increase the speed of localization microscopy by just acquiring the required camera frames in a much shorter time. And finally, we tried to increase the information content per frame and therefore to reduce the number of frames by activating more flow force per frame. And I will tell you now about all of these approaches. So let us start with automated high throughput super resolution microscopy. So in my group, we built the microscopes ourselves. They look a bit like this, and we built them in a way that they're completely computer controlled. We controlled them with Micromanager, but Micromanager didn't have all the functionality we needed. So we wrote quite a few plugins for Micromanager that then this microscope can acquire one region of interest after the other in a completely automated way and acquire additional data. And also what is quite useful, not only for this automated high throughput microscopy, but also for any kind of measurements we do, is that this Micromanager plugin automatically adjusts the UV laser power to keep the number of activated localizations constant. Because of this, my people can just put the sample and they don't have to sit in front of the microscope and adjust the UV laser power. They can just come back a day later and then have hundreds of regions of interest already acquired. Initially, one problem that we had was that the buffer we need for these measurements, the blinking buffer, went bad. And this is well known that the oxygen scavenger that we use leads to buffer acidification. And after a few hours, typically one has to exchange the buffer. But we kind of optimized this and found that if we use a sufficient volume of the buffer and seal the sample chamber well, this buffer can survive for days. So the final thing we need for this high throughput microscopy is an automated analysis. For this, we have our own software that checks the location on the hard disk. And as soon as new data is written there, it's fitted directly on the fly. So when we come back the next day, we have our hundreds of fields of view already completely analyzed. So if you are interested in any of these parts, you can check out our group homepage. There you find links to the software we wrote, but also to some blueprints of the hardware or the electronics we use for this. So now I want to give you a brief example of what we could do with this automated localization microscopy. And here we were interested in a very important cellular process, which is called clustering-mediated endocytosis. And we investigated this in budding yeast as a model system. So endocytosis is the process by which cells take up molecules from the surface and the environment. It's kind of how cells eat. During endocytosis, more than 50 different proteins in many copies assemble at the plasma membrane, and then within 6 seconds, pull in the membrane and pinch off a vesicle. But endocytosis is also quite difficult to study because of this huge complexity, the fast dynamics, and the small size, way below the resolution limit of optical microscopy. Because of this, we don't know nearly as much as we would like to where all these proteins are during the endocytic process. And this is what we wanted to change using super resolution microscopy. And indeed, we used our automated microscopes to acquire images of hundreds of thousands of images of endocytic structures. And again, these are really huge numbers. This corresponds to many months of uninterrupted microscopy time around the clock. From these data, we could reconstruct the average distribution of 23 different proteins involved in endocytosis. This is about half of the proteins. What is nice to see is that the proteins form really very different organizations during endocytosis, but also that the structure is related to their function. So cold proteins form patches of different sizes, actin-regulating proteins form rings of different sizes, and actin-interacting proteins form something in between rings and patches. From these data, we could now deduce a more refined model of how the endocytic machinery is structurally organized and how it is assembling. I really don't have time to go into detail here. I just want to point out one discovery that we made here. This is how the force is generated and transferred onto the membrane. So it was known before that it is the actin machinery that provides the force during endocytosis in yeast. So we now found that one protein, the so-called LAS7 protein, which is a wasp protein, forms a ring at the endocytic sites. Wasp is required to branch new actin filaments off the old actin filaments. And that means we can only have a growth of this actin network where we have wasp. Because of this, new actin can be only added below the old actin. And this pushes up the whole actin structure. And since the actin is tethered to the membrane, this just pulls in the membrane through the center of the ring. So now we are very interested in understanding how precisely this happens, how all these proteins come together to perform this action. And of course, ideally, we would take a 50-color super resolution movie with maybe 10 milliseconds temporal resolution in the living cell. But unfortunately, this is not yet possible. So our approach is to instead reconstruct these dynamics from individual images taken in fixed cells. Again, we use our high throughput super resolution approach to acquire thousands of high quality dual color images. Then we use features in these images to sort them according to time, and also to align them in space and to combine data sets of different proteins in a composite data set. So quite nice way of showing such data is to run them as a movie, but I have to stress, this is not a movie, this is a reconstruction taken from fixed data. So this proof of concept movie here shows the plasma membrane, this protein LAS17 I've been talking about, and an actin binder, and it runs in real time. And it would be quite difficult to get a similar spatial or temporal resolution currently with any other live cell super resolution technique, even in one color. So now we are, of course, in the process of adding more and more proteins to this dynamic reconstruction, because this would directly tell us where they are during the endocytic process and give us a lot of information about their function. So clearly, high throughput or automated microscopy can be very useful because having your microscopes run around the clock produces an immense amount of data. However, it doesn't help us with the second point, the live cell localization microscopy, because we could not increase the speed of a single measurement. So for this, we now tried what actually many groups tried before us, and that is to just increase the imaging speed, to take the required frames in a much shorter time by using faster cameras and higher laser powers. So we wanted to investigate this approach systematically. So we started with our standard conditions. Here you see our nuclear pores that we used as an assay. And here we used exposure times of 30 milliseconds and a laser excitation power by which the fluorophores stayed in the on state for about 1 to 2 frames. Now we systematically changed the exposure time and the laser power in a way that the product of both stays constant, because this leads to a constant light dose per frame. And in an ideal world, and this is of course a linear world, this should not change anything in the results. But what do we see? Well, we see actually quite dramatic changes. When we go very fast, we see that we lose a lot of localizations, and the localizations that we keep are not very well defined anymore. On the other hand, if we go very slow, we seem to get even prettier images than under our standard conditions. So we now could use our nuclear pores to quantify this. And we found that indeed, if we go with very high laser powers here and go very fast, we lose about 2/3 of our fluorophores. We only see 1/3 of the fluorophores that we have in the sample. If you look at the number of photons we detect per localization, the difference is even higher. We see here that for the very slow imaging conditions, we obtain about 10 times as many photons per localization as if we go very fast. And this directly translates in a difference in localization precision or resolution of a factor of 3 between slow and fast imaging. And a factor of 3 is really quite substantial in the field of localization microscopy. So to summarize this, we found there's a clear trade-off between speed and data quality in single molecule localization microscopy. And if you can afford it, if you have the time and want to have the highest quality, we suggest to use rather low laser powers and longer exposure times. When we really drove it to the limit here, when we chose exposure times of half a second, then we achieved a quite amazing resolution. This is here 3D data where we see the 2 rings, and in each of these rings, we can basically almost resolve any single protein. There's something else that we kind of found out when we were doing all these experiments, and that I think can be quite useful for many people who redo localization microscopy. And this is how to switch off the fluorophores at the beginning of the experiment. Because the fluorophore, if you use, let's say, organic dyes for localization microscopy, then at the beginning of the experiment, they're all in their bright state. And in order to then image them in the single molecule blinking mode, we have to transfer them to the dark state. And usually this is done by illuminating the sample the first couple of seconds with a high intensity of the red laser. So we now investigated this and found that during this initial off-switching step with high laser powers, we already bleach and destroy half of our fluorophores. So when we start our actual experiment, only half of our fluorophores are left. We can mitigate it to a quite large extent by using low laser powers instead of high laser powers for this initial off-switching stage. And then even if we perform quite fast imaging later on, we still obtain a quite high labeling efficiency. So to summarize, we found this trade-off between speed and quality. And also we found that it's better to switch off your fluorophores with a low intensity if you want to keep your localizations. So you see what we did here. We wanted to go very fast, but we ended up going even slower than we went before. So clearly this doesn't really solve the problem we have with going to live cell localization microscopy. So as a last point, we tried out to just increase the number of localizations per frame to acquire the same number of localizations just using fewer frames. The problem here is if we increase the number of localizations per frame, then we obtain these overlapping point spread functions. The single fluorophores are just too dense to be perfectly separated. And this is a problem if you use our standard fitting approaches, which assume that we look at single isolated emitters, and they then completely fail, as you see here. So these are simulated data. The cross is here the real position of the fluorophore, and the red circles are the fitted positions. Of course, people try to improve on this, and and many groups are now developing so-called multi-emitter fitting algorithms that can deal with these situations. Unfortunately, till to date, these multi-emitter fitting algorithms are not very robust when we look at 3-dimensional samples. So in a recent software challenge, and I come back to this, it became clear that even standard single emitter fitters can be better than these multi-emitter fitting approaches when we come to dense 3D structures. So to overcome this problem, we then paired up with computational scientists in Geneva and in Thüringen, and together we wanted to use deep learning or machine learning for single molecule localization. And together we developed this software called Decode that uses convolutional neural networks to precisely determine the positions of fluorophores in high density 3D data. So how well does this work? Well, it turned out to work surprisingly well in terms of robustness and also accuracy. So first we compared our software Decode with other fitting algorithms used in single molecule localization microscopy in this localization microscopy software challenge. And this is a platform where you can download test data. You don't know the ground truth. You fit the test data, uploaded it, and then the web page calculates all kinds of quality matrices. And if you don't know this page, I highly recommend you visiting it. So it turned out that Decode really outperformed any other algorithm in the software challenge on any modality and also on all the different test data sets they were there. So it really performs very well. And this is very nice because it allows us to now push single molecule localization microscopy into a new regime, the high density regime. And this we can use, as I show you here, to increase the imaging speed. So here we imaged the same region of interest, showing microtubules 4 times. And by using different UV excitation powers, we tuned the density of activated emitters from the standard density here, that's what we usually use, to the high density, as you see here. And with the high density, we could obtain the same number of localizations as for the low density, but in less than 1/10 of the time. So we are really much faster here. And if you now look at the images, the image quality, especially here in the side view, you see that, yes, we lose a little bit of resolution, but the quality is still quite acceptable here in the last case. We then compared it to really one of the best multi-emitter fitting algorithms. It's the C-spline algorithm of Hayes-Babcock, which I must say performs exceptionally well usually, but under these very high density conditions, it does not anymore. So this is out of the range for standard likelihood-based multi-emitter fitting algorithms. But these increased imaging speeds are especially useful if you want to do live-cell super resolution microscopy. And here we could indeed take an image of nuclear pores in the living cell in just 3 seconds. And again, the image quality is not so bad. But there's another big advantage of using this high density activation for live cell microscopy. And this is that it helps us to reduce phototoxicity. Because we don't use higher laser powers, we just activate more fluorophores in a much shorter time. This allows us to reduce the imaging time and with this, the light exposure and phototoxicity. So finally, we can use decode to image samples that previously we could just not image because they were too dense. And I think many people of you who do localization microscopy using even our favorite dye, Alexa 647, sometimes get these samples that are so densely labeled that when you put them on the microscope, even without any UV laser power, they blink so strongly that we cannot see single molecules. And this is because these fluorophores have a tendency to go back into the bright state spontaneously or activated by the red laser itself. What we would usually do for these samples is to use a pre-bleaching step. That is to really hit the sample for many minutes with a high red laser power, maybe with some UV laser power, until the fluorophores are bleached and we reach the single molecule blinking regime. But then we end up again with these kind of sparsely labeled structures, which we don't show so many details as we would like to. Now with decode, we can image these samples from the beginning and indeed reach much higher labeling densities than after pre-bleaching. And as you see here in the side view reconstructions, if you compare them, we really can see additional features due to this strong labeling. For instance, this hollow cylinder-like structure here on the left-hand side, that is difficult to see if we only see the single fluorophores. So altogether, I'm quite excited about this new approach, and we already made it available to you guys. So if you're interested, you can just go to the GitHub page. Also, in a few days, there is this workshop, the virtual conference, where we have a workshop on decode. I think that the registration deadline is already over, but all of these workshops are recorded. So in a few days, you will be able to get a nice introduction in decode online if you like. So to summarize this part, so we could indeed increase the throughput and speed of localization microscopy. I think to go high throughput and to automate the microscopes as much as possible is a nice approach because it allows your people to not spend hours and hours or days in front of the microscope, but spend time maybe on data analysis or on sample preparation. But if you want to go really fast, our preferred option at the moment is to leave the single molecule regime and to go to medium density and then use these new fitting algorithms. So with this, I'm at the end of the talk, and I want to end by, of course, thanking all the people involved in these projects. Most really my group, my fantastic group here at the EMBL. It's always a great pleasure to work with all of these really talented scientists together. But also we have really great collaborators at the EMBL and outside, and I want to acknowledge the funding that we get. So thank you for your attention, for listening, and I think we can start with a few questions.
Jonas, thank you very much for a fabulous talk. We have already a few questions. So to respect time, maybe we'll ask one or two, and then the rest of them we'll move to the Meet the Speakers session.
Yeah,
So
gladly.
the first question we have is by Amir Hamani. In multicolor single molecule localization microscopy, you use different dyes, and each of them has a different photon flux in the on state. And this can make a difference in localization precision for each channel. Have you observed this difference as an obstacle factor? And if yes, how did you circumvent it?
This is a very good point, a very good observation. Of course, not all fluorophores perform as good as the others. And if it was already kind of work to find 4 fluorophores that are compatible with each other, where the spectra are far enough apart and that work under the same blinking conditions. And indeed, not all of them have the localization precision. Some have maybe 2 or 3 times higher than the others. Some have a switch on and off better than others. This is not a big problem. Then you just get one color with a slightly lower resolution. And you then also have to choose your colors wisely. That, for instance, if you have low abundant proteins, you might label them with the fluorophores that switch on and off more time. If you have proteins where you really want to know the position precisely, you label them with your best fluorophore.
And now a second question from Veronica Labrador. So she asks exactly how you estimate resolution increase in your methods. And she mentions a reference where some beads are used. So regarding, I presume this is regarding nuclear pores. How do you estimate resolution and how it compares with other methods?
Yeah, this is a very important point. How do we measure resolution? And I think it's also a point that has not really been solved. So one way we can give some kind of number to the resolution is by looking at the number of photons we detect, by knowing the shape of the Poinsett function, by knowing the background, and calculate the best possible resolution under these conditions. It's called a Kramer-Rao lower bound, and this is the localization precision that people often report. This is kind of a best case scenario, but it still is, I find it quite useful because it allows us to compare different conditions. But if you have additional problems, let's say drift and vibrations, then of course the real resolution will be lower. And to measure this is not so easy. So one possibility is to draw lines across your structures, for instance, the corners of the nuclear pore complexes, and look at the width of these structures. But one has to be very careful not to do cherry picking and so on. There are many problems connected with this, but this is one way of doing it. The other way is to use something, some methods that have been developed, such as Fourier ring correlation. Again, this is very useful. We use it a lot, but also this has some drawbacks. For instance, it's very difficult for this algorithm to distinguish a single fluorophore which comes back many, many times and therefore might appear as a high resolution from kind of the real resolution that you have in your super resolution images. For using beads, I'm a bit skeptical because this is always very, very far away from your real measurements. For example, if you tune their excitation power that the beads have a brightness similar to your fluorophores that you later use, you end up with basically no background noise, and you greatly overestimate the resolution that you would get using single fluorophores.